Enzymes are able to distinguish between alpha and beta glycosidic bonds. So when we name a glycosidic bond, we specify whether it's an alpha or beta glycosidic bond. And that alpha or beta distinction depends on whether the anomeric carbon to oxygen bond is above the plane or on the same side as C6 or below the plane of the ring or opposite side of C6. And when two monosaccharides bond together, it's also important to identify which carbons the hydroxyl came from that reacted, because monosaccharides have a lot of different hydroxyl groups. So for example, for maltose, we would call that an alpha, and then in parentheses, 1 arrow 4. So the first carbon that is always listed is the anomeric carbon, and in glucose, that anomeric carbon is always C1. And we didn't really pay too much attention to the molecule that the second molecule in this disaccharide. It's also D-glucose, 1, 2, 3, 4. The hydroxyl group that reacted with the anomeric hydroxyl group was on the 4 carbon. So that's why we called it an alpha, parentheses, 1 arrow 4 glycosidic bond. Now, so that's how maltose, which is a disaccharide, is connected. If we look at cellobiose, which is also made of 2 glucose units, but these are made from beta glucose molecules. So the hydroxyl group was originally above the plane of the ring structure. And so we see with the anomeric carbon bonded to this oxygen in the glycosidic bond, it is above the plane. So this is beta 1,4 glycosidic bond. The hydroxyl that the anomeric carbon reacted with came from the 4 carbon on the other glucose molecule. So these two may look very similar to us, but actually they are incredibly different to enzymes. So humans do not have the enzymes necessary to clip the beta glycosidic bonds. Whenever we have disaccharides or polysaccharides or oligosaccharides, it is necessary for us to be able to break down that glycosidic bond because it is actually glucose that our cells use for energy. So if we cannot break this beta 1,4 glycosidic bond, we cannot use anything that has cellobiose for energy. So cellobiose is the major structural component of cellulose, which is a polysaccharide made of glucose, but it is comprised of those beta 1,4 glycosidic bonds. So some animals, such as cattle, deer, giraffes, and cattle, can digest cellulose, which is found in leaves and branches and twigs, because in their rumen, which can be considered one of their stomachs, they have bacteria which secrete an enzyme known as cellulase, and that cellulase is able to break down the beta 1,4 glycosidic bond. Termites also have bacteria in their digestive tract that secrete cellulase, so that's why termites are able to digest wood fibers, which are made up of cellulose.